I am pleased to be hosting Dr. Fernando Lopes and Dr. Conrad Gillett to discuss the recent PLOS ONE Lab Protocol paper entitled From Museum Drawer to Tree: Historical DNA Phylogenomics Clarifies the Systematics of Rare Dung Beetles from Museum Collections, and the associated detailed protocol in protocols.io. Fernando is a postdoctoral researcher at the State University of Campinas in Brazil. In his current research, he uses genomic data to explore the evolutionary history of the Peruvian fur seal, a hybrid lineage, examining the impact of climate change on the secondary contact with one of its parental species and the implications of this phenomenon for the species' future. Previously, he was a postdoctoral researcher at the Finnish Museum of Natural History of the University of Helsinki in Finland, where he studied the phylogenetic relationships of scarab beetles from Madagascar and surrounding islands using genomics and museomics techniques. Conrad is a researcher and curator in the entomology team at the Finnish Museum of Natural History, specializing in systematics, evolution, and biodiversity of beetles. His work combines molecular and morphological methods with a focus on scarab and ground beetles. He co-curates the entomology collection of the Finnish Museum of Natural History and teaches at the University of Helsinki. With a lifelong passion for beetles, Conrad has worked in museums worldwide, including the Natural History Museum in London, the National Zoological Collections of Suriname, and the University of Hawaii Insect Museum. Fernando, Conrad, thank you so much for sparing some time to talk to me.
Thank you very much for inviting me.
Yeah.
So to kick off, can either of you briefly explain what this protocol is about, how to use it, and why it is relevant?
I think I can start with it, Conrad, and then you can complement if you feel comfortable to do it. The protocol was originally developed to recover highly degraded DNA from vertebrate specimens in museum wet collections. So basically, we retained its core methodology, but we made minor adjustments to optimize it for insect-like collections, specifically speaking, dung beetles. Well, Conrad can say it better, but insects, they represent the most diverse group of organisms on Earth, yet their abundance and diversity remain vastly understudied. So entomologists, they rely heavily on museum collections. We are critical hubs for biological research and where specimens collected over centuries are preserved. So these archives, they are foundational to biological sciences and host irreplaceable genetic and morphological data. So by adapting this protocol, we help it unlock the potential to extract DNA from historically preserved insects specimens without completely destroying them, like using non-invasive methods. This enabled us to make new insights in biodiversity and evolutionary histories of these groups that would otherwise remain in the shadows.
Yeah, so I'd just like to highlight the fact that insects and beetles in particular are the most biodiverse organisms on Earth, yet they remain vastly understudied, of course, because there are so many of them. But many species are, in fact, rare and represented, in many cases, only by old museum specimens sitting in the various natural history museums around the world. And so, therefore, if you want to study the diversity and evolution of these organisms, inevitably you end up needing to look at the study museum collections, and that the stumbling block has been that the older the specimen, the more degraded the DNA is, and the more difficult it is to access that genomic information. So this protocol has contributed to enabling access to that genetic data from these old specimens.
Even with not knowing the history of the specimens, right, Conrad? How they were collected, how they were preserved over time.
Yes, that's right, because that's very important too, because well, we don't know how some of these older specimens were collected. They almost certainly were not collected. Well, the intent of genetic research to be undertaken from them, and in fact, it's remarkable. I think it's remarkable to think that the people who collected these specimens in the past, they could not have known, they could not have imagined how those specimens would have been used in the future. In some cases, specimens that were collected even before DNA was known as the hereditary material can be used nowadays in genomic studies. And I think that really highlights the importance of museum collections as this vast repository of biological genetic information. And these advances in laboratory methods and bioinformatics methods too are unlocking the potential that these specimens hold.
And if I understood the paper correctly, the base for your optimized protocol was a previous protocol that was optimized for wet collection specimens. Can you elaborate a little bit more on what optimizations you had to implement to have this very specific protocol work for insects that might have been preserved in different conditions?
Now, this is a quite curious story, because actually it started from a failure project that we have. Everything started actually when I went to the University of Bergen to join the Dr. Nicholas Straub group at University of Bergen. He had previously developed this protocol for extracting degraded DNA from vertebrate museum wet collections, as you mentioned. So I traveled to Norway to apply this protocol to subfossil dung beetle samples. They were kind of 400 to 2,000, 4,000 years old samples. They were from Madagascar islands, but unfortunately the results were unsuccessful, mainly because of the nature where the samples were collected from, tropical and humid environments. So no usable DNA was retrieved from these samples using this extraction method and library preparation specifically tailored for museums techniques or ancient DNA. So back in Finland, to the Finnish Museum of Natural History, my supervisor and I, we had this breakthrough. If the protocol worked for vertebrates, why not for insects? We have tried with subfossil samples, but why not with some kind of better preserved samples? But unlike vertebrates, which often have abundant tissue and bone samples for DNA extraction, insects, they present a unique challenge due to their smaller size and more fragile remains. So we then invited Conrad for this project because he is a beetle specialist. He has interest in museums during this project, and then we invited him to join us. So at the time, we were studying the systematics of dung beetles from Madagascar and Mascarene Islands, but exclusively using freshly collected samples that we had collected in the field in Madagascar. So for this work, we shifted our focus to historical samples by also incorporating enigmatic samples and from species from Madagascar, Mauritius, and Thailand that were preserved in 3 different museum collections: the Paris Museum of Natural History, the Natural History Museum of London, and also the Finnish Museum of Natural History. The key question actually for this project was if the protocol was developed for these for the wet collections of vertebrates, it was originally developed for this kind of samples, and it was working actually using shotgun sequencing. Could it be adapted for beetles and combined with a newly developed probe set to recover fragmented DNA from these historical samples? I think this was our starting point with this project. And at the end, it worked exceptionally well. So now we can determine the evolutionary relationships of species collected in 40s, 60s, and 80s, specimens that were never, as Conrad mentioned before, prepared or preserved for DNA analysis.
Yeah, and one important aspect of the protocol is that it is remarkably economical, cost-effective, and accessible compared to previously available methods. So this has really helped to make the existing insect collections in museums available for study. And like I said before, it's really remarkable to think that these old specimens are now being able to use in a manner that was previously unimaginable. So yeah, it's being able to access this genetic data that from fragmented, highly fragmented DNA preserved in these specimens. It enables people like me who are interested in the evolutionary history of these species to incorporate more specimens in the phylogenies, these rare specimens that we can't just go out and collect them, because in many cases, well, in many cases we don't even know exactly where they're from, or they're incredibly rare, and it's highly unlikely that you may be able to find them again. So we must resort to using the existing specimens in collections.
I would say that on top of everything is a non-invasive museomics method, like right, Conrad? In many cases, we preserve the whole morphology of the specimen while extracting the DNA that was never possible before. So I would say that in some specific points, the specimen got better after extraction because it like cleaned it up in the end, and you got a bright new and clean specimen.
Yeah, this aspect is this aspect is very important for anyone working in as curators in museums, in that although of course we want the specimens to be used for scientific research, we must always keep one eye on preserving the integrity of the specimens for future scientists to study, especially the morphology. And so, whereas in the past, one may have needed to grind up the specimen, destroy it, in order to extract the necessary DNA from it. It's now, been shown, and our work has contributed to that. In fact, this is not necessary, and the integrity of the morphology of the specimen can be maintained.
And in addition to the cost, which is lower for your protocol, what are the technical differences between the protocol that you successfully put together and those that already existed, and some of which actually failed in your hands, Fernando?
I think, well, the difference from the original protocol is not very huge. We basically used the same thing, but the way we treated the sample was the most important thing in this case. So as I mentioned before, vertebrates, they have a lot of vast tissues and bones that you can use to extract a lot of DNA. If you don't get DNA from one piece of bone or from a piece of tissue, we can take more sample and try to get DNA from it. Well, unlikely, the insects, it's not like that, right? We don't have many samples. Sometimes we can try and retry to extract the DNA. Sometimes we have only one specimen that was stored in one museum abroad, and you have to try to get DNA from this unique specimen. Otherwise, we will never have DNA information from this specimen. I think the protocol, as I mentioned before as well, we kept the core methodology, which was basically the same. Of course, we had to do some adjustments in the protocol, but not it's not something significant, right? Significant. We had to just do some minor modifications. But the most important thing here is how we treated the sample. This was, I think, one of the most challenge thing doing this protocol.
Would that be then the most critical step, treating the treatment of the sample?
I would say so, because sometimes we had to deal with extremely rare and replaceable specimens. And in that case, any damage or loss could be irreversible. For example, I like to cite this specific question, this specific point. We were working with, for example, with Mauritian dung beetle holotype, all right, which is the single specimen that the original species description was based on. Conrad can stress it better. It was from the Natural History Museum of London. It was collected in 1940, representing a species that has not been seen again since its description. So the extraction process was particularly tricky and required precise adjustments to recover sufficient DNA while minimizing damage or any kind of harm to the specimen. So additionally to everything, of course, contamination is always an issue when doing museomics or working with old and ancient DNA. So dealing with contamination or cross-contamination in this case was also crucial in our protocol. So, but for that we worked in dedicated clean room with stereo equipment for that, following all the good laboratory practices for working with museomics techniques.
And I would like us now to talk a little bit about potential negative data. As you were optimizing the protocol and making these changes, was there anything that you tried that failed and didn't make it to the final version of the protocol?
Yes, of course, not the protocol itself, but as I mentioned before, contamination is always a risk in doing this kind of stuff, and we ended up with some contamination in our analysis. For example, we used some positive controls in this case, specimens that we knew the phylogenetic position in the tree of life. By recovering the DNA and sequencing them, we recovered very strange relationships in the tree. So we were pretty aware that it was not something new to science or new to dung beetles, right? It was probably a contamination or something that happened on the way to extract and sequencing those DNAs.
So the protocol was optimized and used in beetles, but would it work in other insects? And would it need any modifications for it to be transferable?
Sure, it would work, of course. However, some modifications may be needed. For example, insects are quite variable. They can have different forms and sizes and colors and so on. So variations may be an issue when using this protocol. So the one trying to use this protocol must be aware of that and must adapt for the protocol. Also, degradation levels might necessitate some adjustments. We encourage others to tailor the protocol to their unique research context as well.
Yeah, in the context of insects, beetles are rather robust. If one thinks of a beetle, you imagine this thing with a hard, external carapace, and many other insects are more fragile than than beetles, and may require more delicate treatment in the extraction step. Yeah, this is
for
like
example,
Fernando says, this is something that people working with those groups will need to optimize if they're trying to preserve the integrity of the specimens.
Yeah, for some specimens, for example, millimetric beetles, we would have to use the entire specimen for the extraction. But for some larger specimens, maybe by removing some muscles or internal content would be enough to extract DNA.
And I would like us now to change gears a little bit and discuss about your motivations to send the protocol to PLOS ONE to be peer-reviewed as a standalone methodological paper.
This is also quite curious because it was first submitted as a research article, but it's a mix of research article and methodological paper. And after the first submission, it was the academic editor's suggestions to submit as a lab protocol. And we accepted, and yeah, there it is as a lab protocol paper.
And how did the feedback that you received from the peer reviewers shape the final version of the protocol?
Actually, it was not, it's also curious, it was not mentioned much by the reviewers, but we think they like it as it is. They focused more on the phylogenetic taxonomic content implications of the results and how it would imply the results and we obtained using the protocol. I think this was previously developed for vertebrates and before that for ancient DNA samples. I think it's quite established in literature and people, they were using it a lot. So there's not much to comment on that. So they focused more on the taxonomy and systematics part of the paper.
Yeah, although I think it's always more interesting if such an article promoting a new protocol has some real world scientific analysis behind it, and you know, we're able to demonstrate that the procedure works well and is useful for anyone working in the museomics field.
And my last question is probably, or the answer to my last question is probably speculative at this time because your protocol is your paper is relatively young. It was published just at the end of last year. But what do you think has been or will be the significance of this lab protocol paper for both of your careers, considering potentially, different stages for each of you?
Well, for me, having this interest in the evolution of beetles, one limiting factor has always been the incorporation of rare species in molecular phylogenies that I construct with my colleagues. This taxonomic bottleneck can really affect the robustness of results. And usually the reason for that bottleneck is simply because existing specimens were considered too old to be useful in terms of providing good DNA sequences. So by establishing that we can actually now in a fairly straightforward manner obtain this genetic data, it's really going to help people like me become more confident in being able to access this information. And it's also good that we can demonstrate to museum curators who may be understandably worried about their specimens being used, their valuable rare type specimens, etc., being used for what would have previously been destructive DNA extraction methods. Well, we can now show them that the specimens are not damaged, or not destroyed, certainly, and their integrity is there for future generations. Because of course we must always think that that the technology is advanced and we want to maintain those specimens for study for the future, just as they were preserved, so that we can study them now. We need to make sure that that we're not going to destroy everything so that they're not available in the future.
Well, in my case, it worked for example, this helped me to grow professionally because it expanded my skills a lot. I'm not a beetle specialist like Conrad, and I'm basically a bioinformatician and evolutionary biologist, which work with genomics in general. And I have experience with many other methods like whole genome sequencing, ddRED, UCEs. And this was the first time that I had the opportunity to work with museomics techniques. So it's a great opportunity to grow up professionally speaking. And it has already given me some how can I say that, some good things, I would say, because I have already applied for some positions in the field of museomics. I have been called for interviews, and it's also great because now I can say that I am more flexible in analyzing data and also working with different types of organisms and so on.
Conrad, Fernando, I don't have any other questions. I don't know if before we finalize it, you have anything to add that hasn't been discussed so far?
I'd just like to add that first of all, I'd like to thank Protocols.io and PLOS ONE for having us here today. It's important to promote even more our manuscript. And anyone that wants to have access to it or wants to learn more about it can contact us at any time. We'll be always available to answer questions about the manuscript and protocol.
Okay, thank you so much.
Thank you.
Thank you very much.